We are very excited to be celebrating the 100th episode of the Immunology Podcast, Viral Immunity, and special guest Dr. Peter Doherty. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Roud. Welcome back to the Immunology Podcast. We have conversations with immunologists. The Immunology Podcast is brought to you by Stem Cell Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave us a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today we have Nobel Laureate Peter Doherty from the University of Melbourne on the podcast to talk about his seminal work on T-cell antigens, MHCs, and infectious diseases. We also got our usual roundup of recent highlights in immunology news coming up, but first...
Don't forget, the registration is open for Immunology 2025. Connect and collaborate in Honolulu, Hawaii from May 3rd through 7th, 2025. Advance your career and access cutting-edge research. There has never been a more important time for the immunology community to gather at the world's leading annual all-immunology meeting. So you can learn more at immunology2025.aai.org.
All right.
How you feeling? I mean, 100. That's 3 digits. This is I'm impressed.
Now you're old too.
Yes, but not as old as you.
Nope.
Never. That will never happen.
Well, if I start running really fast, it could eventually happen.
Maybe. Yeah, that's true. You could, bend space time enough to gain those years.
If it just got really fast.
I wouldn't count on it. I wouldn't count on it though. But yeah, I mean, let's reminisce just for a second here. 100 episodes. That's 4 years. 25 episodes per year. It's impressive. It's like I've been doing this for 4 years.
Does that mean that's like your PhD or whatever, right?
Right. Yeah, it's almost as long as my PhD. It's getting there. Hopefully, we'll surpass it in length. But yeah, it's like what, a bachelor's degree over there or something. Pretty neat.
Yeah, bachelor's 4 degrees too.
Yeah. So yeah, and I don't know what do you think we'll be doing? In the 200 for the 200th episode. That's should start planning.
Maybe we could do it from space with Arun and do a combo like immune stem cells in space at the IIS.
That would be nice. Wouldn't that be nice? Yeah. That's it. That's that we should go for. Immunostem cells in space. So we have like this big mashup with our friends at Stem Cell Podcast. All right. I mean, dear listeners, if you have any suggestions on where should we already start planning for, for our 200th episode, please. I can imagine some comments are going to be something along the lines of, let's just plan a huge LARP organized by Jason. We can dress up as our favorite immune cells.
I could do a LARP for immune cells. We could have some invaders and then I could do this. We can make it work.
Okay. All right. Then start planning for it.
Well, Brenda, do you know why I mentioned space?
Why?
Because I have a paper on space.
No way.
Oh yeah.
All right. Okay. You have my attention.
Space bugs.
Bugs.
No, not quite. All right. Kind of, sort of. All right. So we have, it is in Cell. Comes out February 27th. I'm going to give the name of it last because I want to make you in suspense. First authors, Rodolfo A. Saladino and Hoekui Nin Zhao. Last corresponding authors are Pieter Dorstein and Rob Knight. It is the International Space Station has a unique and extreme microbial and chemical environment driven by use patterns.
Okay. So they're going to talk about, the bacteria in the International Space Station.
Yeah, and the chemical patterns too.
Okay.
They basically did spatial mapping of multiple rooms of the IIS and everything else. Swabs and did, bacterial sequencing and chemical mass spec analysis. And here's what they found. Super high level, it's an extreme environment. If you compare it to human environments, how much is human origin in an environment, like outside or inside or in a house, the space station is unshockingly an extreme high human relevant, no other species environment more than anything else they usually see. This is also true of chemicals. They can see the chemicals used in cleaning and other stuff and in grease and other things on there, but they also see bacterial products. They also interestingly note less diversity than they usually anticipate, unsurprisingly. And where humans are the most bugs, and where hygiene occurs, has even more bacteria than other places.
Those space toilets are not great, I guess.
Space hand washing, space toothbrushing. So it's a really cool paper. Like, they have all these pretty pictures of 3D scans and maps of like where they find urine next to the toilet.
That's the kind of thing. I mean, you can't
Where
just
they
do that
find
with like
bacteria in
a
the oral cavity next to the sink.
UV light. Just to
Yeah,
put
it's
you know, like a
the sequencing heat maps all over the place, and it's really fascinating. It's I nerded out reading this. Immunology related, right? It's really about the bugs and what they can do to people and how you map stuff. But it is fascinating in terms of just what they're able to find. Staph is like the most abundant organism on there. Staph aureus from all our skin.
right.
And humans are filthy and carry a bunch of bugs with us.
Well, is that surprising? I mean, I wonder, did they test? You know, you have like the American part, the Russian
Yeah.
part of the
Yes. They absolutely compared, and
and
they see a little bit of regionality, but people move across them, so not too much. It's mostly about where they lived.
Okay. Well, maybe there's some, vodka specific microbes or like some borscht microbes on one side and hamburger microbes on the other. I don't know.
Yeah.
The freedom microbes on one side and
And the
anyway.
sushi microbes on the other.
Okay. And is there like anything related to, if you're thinking about space and space exploration and is there any
They're worried about a lack
worries?
of diversity, or like
Yeah.
one person has MRSA, then it's just going to spread everywhere.
Right.
And
Yeah.
they look at like, how homogeneous things are, which it's pretty homogeneous, right? There's not a lot of diversity. Like, that staff becomes dominant staff.
Right. Is the microbiome of like this the astronauts kind of starts kind of converging to something if they spend it long enough in the
I don't
space
think they did it
station?
in this study, but yes, it's all
I mean,
based on who you're with.
right? These poor two guys are still there since what was it? June,
I don't
July
know.
last year or something.
I know, it's sadness.
They should study their microbiome and see how different. And how about radiation? Like
Well,
how
they're
protect?
radiation protected, right? Because if the radiation was getting you, it'd cook the human. So
Right.
everything's hardened.
Well, but they get some radiation more than
They
on Earth.
do. They didn't look that with the bacteria, but they don't get enough to really jack you up. Because if you were mutating all your E. coli all the time,
Yeah.
you would have a problem.
Exactly. That's what I was thinking about, some nice mutant cells. Exactly. Imagine this new kind of ultra resistant E. coli that comes with the astronauts in the next return home.
Yeah.
I mean, it's not that we could be any worse nowadays for the world. So that would be the cherry on top for our current global instability. All right. Uh Fascinating. Microbes in space. You know what is also fascinating?
What? Besides space?
Yeah, well, that's a tough one. You really start very strong.
I didn't.
Well, but you know what? What I find that I get really, uh, really captures my imagination and my attention when you have beautifully designed studies with really nice experiments and really cool results. Then also, it inspires me. So let's talk about my first choice of today, and which I thought was such a nice piece of work. So let's dive into it. So the paper I'm going to talk was published in Science. I think it was February 27th, first release. First authors David Clawan and Nicole Pagan. From corresponding authors Peter Savage, um, Harikesh Wong, and Roland Germain, who are at MIT, at the University of Chicago, and at the NIH. And the paper is called Regulatory T cells constrain T cells of shared specificity to enforce tolerance during infection. And I know, I gathered, T-Reg paper. How unusual of me. How biased I am in my selection. But I really like this publication because, I think I mentioned it before, and it's actually part of my current research in my day job as a postdoctoral survivor, in which it's a, we know that Tregs, this C4 regulatory T cells, these cells that express FOXP3, and we know they're really important. If we don't have Tregs, uh, things get very, very difficult very quick. Mice that lack functional Tregs, they get autoimmunity, off the bat. Humans that are born with, deficiencies in their Tregs, mutations in FOXP3 and such, they don't survive very long if they don't get a bone marrow transplant to correct for this deficit. So we know for a fact that Tregs are capable of kind of establishing a dominant suppression of those T cell clones that for some reason in the periphery are present that can recognize self peptide. And there's always this um it's not completely clear, how this works. And the traditional view is that you have clonal deletion of conventional cells during thymus development, because self peptides are presented on thymus epithelial cells, and these get, if you have a T cell that is activated, uh, when it's presented with a self peptide in the thymus, it either deletes out of the repertoire or becomes a Treg. That's kind of the classical view. But then the truth is that then you see that in the periphery, self-reactive T cells that are not Tregs exist, and they become problematic, and then you have autoimmunity. So they in this paper is really cool because they use a mouse model in which there's a specific peptide that is derived from a protein that is expressed on the prostate. And so you have in male mice, you can find Tregs that are recognizing this very specific peptide called C4 in and these Tregs accumulate in the draining lymph node that drains from the prostate. So they're activated. They are there, and that's how they identified this important kind of antigen that is specific to the prostate. Females don't have a prostate, but they do present the peptide in their thymus because of air and all this very kind of promiscuous expression of genes, and therefore they still have Tregs, but they don't accumulate in the draining lymph node because there's no prostate to drain from, kind of. So this really shows the idea that Tregs can recognize this peptide. And similarly, you can have conventional cells recognizing this peptide too, and if they get activated and they proliferate, basically you have prostatitis and you have inflammation in the prostate. So when they look into so they have these T cells. If you transfer T cells that are specific for this prostate antigen, and you transfer into wild type recipient mice and male mice that have a prostate, what's interesting is that they these CD4 cells will get activated, and this is measured by activation markers downstream of TCR signaling, and they will proliferate, or we try to proliferate a bit, but this is short-lived. And if you don't have like a kind of other inflammation, basically, although T cells will recognize the antigen, they don't get to form a response that results in inflammation of the prostate. And this is kind of conceptually important because this is the idea that there is something else, like there's all these other mechanisms that prevent self-reactivity. On the one hand, you need kind of some level of kind of co-stimulation, and but on the other hand, you have the Tregs that are preventing active responses. But this only so what the authors looked into is like how exactly what matters when. So if instead under steady state these CD4 cells they find their antigen but they don't get activated, they don't initiate a full response. If you remove the Tregs by knocking out, for example, using a diphtheria toxin receptor that will remove Tregs from the mice, this is still not enough to uh, initiate a reactive response. So it is not. It is there is still like this indicates that although Tregs they do when you have activated conventional cells, they what they find is that they get enriched around these cells that are becoming that are sensing something. They have signs of T cell stimulation, and Tregs they kind of seem to be kind of establishing themselves around these cells and kind of keeping them from activating further. You still have kind of this initial inflammation that is required to get all the right signaling. So what do? They take a Listeria monocytogenes, which is a very commonly used, infectious agent for many studies. And what is cool is that they include this Listeria that will also contain the C4 peptide that is the one that is recognized. And so when they actually infect the mice with the Listeria with the C4 peptide, this is sufficient to activate conventional cells, but it will only lead to an actual, inflammatory response in the prostate and actually get a proper activation when there are no Tregs, when there are no C4 specific Tregs around. And they do this because they delete in the thymus exclusively the C4 peptide, so it can never be presented. So these mice never have Tregs, but they do have conventional cells. And therefore, they find that under a very inflammatory and with a lot of antigen, which is what happens when Listeria starts infecting the mice, then this is sufficient to break the tolerance, and that's where the Tregs become very important. And if you remove the Tregs as well, then you have inflammation of the prostate and inflammation, and you have an autoimmune response. So they really kind of play around this idea, and the idea that is very important. The specificity is very important because when they look at, even though the Tregs can inhibit a T cell response against this particular peptide, the C4 peptide, it does not affect the response against other Listeria peptides. So really showing that it's very specific. The specificity of the Tregs is really directed against the peptide they recognize. And there's sometimes little kind of cross regulation. So having Tregs that protect your own antigens is not in detriment of the immune response against an infection, for example. They show this kind of by doing this following the immune response through time, and they show that basically Tregs can restrict the emergence of these fully activated C4 specific T cells, and they do this in part because they seem to activate quicker. They're quicker to activate, and this is by a few days, very like a day or two. But this is enough to give them the edge to kind of, once they get activated and they find themselves in the kind of in the close to the activated conventional cells. That's how they prevent the conventional cells from activating further and mounting a full response. So basically, you see that the conventional cells, they have an initial small uptick in their proliferation, and they measure with Ki-67 expression. And then if Tregs are present, this becomes, they kind of ebbs away. But if Tregs are not present, this kind of builds on, and you start seeing actual proliferation and increasing the numbers of the conventional cells. And what's important again is that they don't seem that the presence of the Tregs doesn't seem to be inducing energy or like a functional kind of some kind of irreversible energetic state on the conventional cell. They just don't get to proliferate, but they don't seem to show signs of energy or of loss of function. They just don't get to proliferate, and then they just go back to kind of baseline. And this is what the Tregs are doing by being there. And again, this is only happens when you have both the conventional and the Treg recognizing the same peptide. And so they also do some analysis when they have fused peptides. They can be presented by the same dendritic cell, but still somehow you see that the responses against one peptide seem to be kind of separated physically in some dendritic cells focused on one peptide, and that the other dendritic cells seem to be presenting the peptide that is in the same fusion protein, but there seems to be some kind of spatial separation that really allows the Tregs to only inhibit their response against the self peptide that they're supposed to be protecting, and not other peptides that might actually be uh, coming from the infectious agent that the dendritic cells are presenting. So I thought it was really nice. So this paper was a lot of work, but basically shows how important that although Tregs are self-specific, Tregs seem to be dispensable at homeostasis. They are really important to selectively suppress self-reactive conventional cells while allowing pathogen-specific responses to continue, and that they do this by forming. They do this with microscopy to show they form a kind of localized microenvironments, and they surround physically and functionally constrain conventional cells from mounting a full activation response. And they seem to have a edge in their speed by which they get activated, and that's how they can kind of be one step ahead of the conventional cells and prevent them from continuing with their activation for long enough to establish an immune response. And it's all done in a beautiful mouse model.
So do you think it's human relevant?
Yes, I think so. Yeah. I think so because there's other examples. Of course, you don't have human models, but there's examples of antigen specificity in certain diseases where you really know which are the peptides that are, mediating the response. And you do see that, for example, in diabetes, you do have Tregs that are, so some people have Tregs that can recognize these peptides that are targeted in type 1 diabetes and patients that don't. And this seems to correlate with whether they actually have destruction of the pancreas or not. So the presence of these Tregs seems to be protective. Correlation, it's not causation, but I do think we have some observations in humans that would fit this. That's why I really like this paper.
But I thought we already know Tregs constrain T cells.
Yes, but I think the the question is whether we know they constrain, but there is still some uncertainty about how specific that constraint is because you say, well, if we have Tregs that are activated at the same kind of within the same global response, maybe that's enough. And here it shows that it has to be exactly the same peptide or exactly the same recognition because adjacent recognition, so to say, even though Tregs are recognizing, a peptide. We have a protein, and Tregs recognizing one part of the protein are not going to inhibit the activation of conventional cells recognizing a different part of the protein. So it really, I don't think we completely understand what is the kind of the sphere of influence of an activated Treg when it comes to this, and I think this kind of indicates it's very narrow.
Interesting. That's interesting. All right. Well, to keep things moving, I'm going from the one confined space to another here. This paper is in Nature. Last authors contribute equally: Mary Kay Hayden and Julia Segre. First author is Diana Proctor. Coming out 26th of February. It's Clonal Candida auris and Escape Pathogens on the Skin of Residents of Nursing Homes. So, top line, nursing homes are filthy and they transmit disease.
Great.
So, more interesting, this is a study of the multi-drug resistant human fungal pathogen Candida auris, as well as escape pathogens, that's Enterococcus epicalum, Staph auris, Klebsiella, Actinobacter, Pseudomonas, and Enterobacter species, and then other species such as E. coli, Provencia, Proteus, and Morgella. So the big bad bugs we all look at. Okay, so what's interesting about this is what they looked at is the clonality of it. So one, this was a proof of principle paper where they're showing that you could do sequencing, whole genome sequencing to track bugs, which usually require, like you sometimes issues with read depth, you can't get down a strain. They apply some methodology to be able to do strain analysis pretty easy, better, and be able to show proof of concept. They can just use shotgun, which is cheaper, but you can just do shotgun and everything, right? And showing that show that they can do shotgun and everything and get strain level information. So that's really neat. But then what they show is that there's a lot of clonality. So roommates really share clones of organisms. Surfaces such as hand, they looked at different surfaces, inguinal, perianal, your hands. Hands, very clonal. They actually use perianal region to see how much of this is fecal transfer, and it's not that much, actually. It mostly looks like hand transfer from people touching stuff to each other. And not only that, but like there's just a huge level of like species level clonality or 1 to 2 SNP differences. So just small genetic drift over time within a nursing home of these bugs. And then they can see it even transfer between nursing homes in the same city. And they've even seen it transfer from a known case from, I think, New York to Chicago of an organism that like a nursing person in New York went to a Chicago nursing home and then transmitted it to that entire nursing home. But interestingly, when they compare to Europe, they don't see transmission internationally of the organisms clonally.
It's a language barrier. That's why.
Yeah. But like, it's just fascinating how, that they could do shotgun sequencing, which is cool, but and they also do orthogonal methods. So they did a bunch of different methods orthogonally to show that it was working to track, which is, I think, part of the proof of concept here. But there's just tons of these pathogens on everyone's skin. And they all share it across each other in a nursing home.
But it's something about the population of nursing home, or if you would expect the same, like, I don't know, kindergarten or something like that, or something about old people.
Something very similar in kindergarten. Now, they may not have the same bugs on them because old people and young people may have different bugs on them. Yeah. I think you expect something similar, less common among office workers, right? Like old people and young people touch their faces and mouths and each other a lot.
All right.
Also, they don't talk about this here, and this is more of a sniff where people are more sick, but regular nursing homes are rampant for STD communication or STI communication.
Okay.
No, it's like really like most outbreaks of gonorrhea and chlamydia are in nursing homes and syphilis these days.
Oh, Christ. Is that because they're having too much fun, or is it something nefarious? Okay.
No. It's for the exact right reason.
All right.
Because they don't have any birth control concerns, so protection goes out the window.
Right. Well,
but they're saying, but not in that way. But they're also just saying hi to each other all the time and playing bridge or having a nurse clean them. They do see that aseptic baths reduce. Part of this is they know aseptic baths reduce it, and they have a little data about that in here too. But fundamentally, like me and my roommate both have the same multi-drug resistant organisms after a little bit of time. And then eventually someone other side nursing has the same one or one that's just mutated slightly with 1 SNP difference. And when I get transferred to the next nursing home nearby, they get it too. Oh boy.
Would this be informative if you wanted, for example, to influence the communal microbiome to something that maybe is less pathogenic or something?
Maybe you just put some bugs that compete off these things in everyone's yogurt every day, and then you can stop the insanity of MDROs. But it's just wild how, just like space, it's an extreme constrained environment. The sniff.
Oh, very nice.
It's nuts. It's like super clonal and kind of scary.
Well, that's exactly what we wanted to know today. I mean, a lot of, bug communities for today's papers, Jason.
I'm here.
We're a little bit monothematic today, if I may say.
I was monoclonal.
There you go. And with that joke, how can I live up to that? I'm not sure. But okay. My paper also has to do a little bit with bacteria. So, I guess it's kind of continuing on the trend. So the paper I'm going to talk about now is called Macrophages Recycled Phagocytosed Bacteria to Fuel Immunometabolic Responses, published in Nature, 26th of February. First author, Juliette Lesbats, from the lab of Johan Garraud, University of Bordeaux. And I'll try to make it, succinct, but I think it's kind of cool. But basically, they show something that, when you think about it, it's not so surprising. Macrophages, the little trash cans of immunity, they, phagocyte stuff, as we all know, and they phagocyte bacteria. And surprisingly, they are capable of eating up that bacteria and using the building blocks of the bacteria as fuel. And also they are influenced by the contents of this bacterial engulfment, and this actually affects some of their kind of metabolism and their phenotype. So basically, what they show is that they start, feeding uh, bone marrow derived macrophages with heat killed Escherichia coli. So basically, dead bacteria, and they compare dead bacteria to LPS, which would be a TLR4 stimulation that is often considered kind of equivalent to stimulating, macrophages with actual bacteria. And they show that actually there's a lot of differences when it comes to the metabolism and certain gene rearrangements that happen when the bacteria are activated with so when the macrophages are activated with dead bacteria versus just LPS. And this all kind of points towards the bacteria, the macrophages actually taking up the bacteria, uh, bring them into a phagosome and degrading the things from the bacteria and incorporating those nutrients from the dead bacteria into their own metabolic activity. So macrophages that eat killed bacteria, they increase glycolysis, oxygen consumption, and this depends on their capacity to kind of acidify a phagosome and degrade bacterial stuff. And they, amongst the things that they take up is amino acids. So they incorporate amino acids they try, labeling bacterial amino acids, and then they see that the labeled bacterial amino acids become part of proteins in the macrophage, and they become part of, metabolites from the Krebs cycle in the mitochondria of the macrophages. So they really are, when you give this killed bacteria, they really are incorporated by the macrophages into their own metabolism. And so one of the, I think what's interesting is they show this in vivo as well, with label macrophages in vivo and mice. But I think what's also really cool is that some of these metabolites that become really fed by bacterial derived nutrients are those included in glutathione and itaconate biosynthesis. And this is important because glutathione is an antioxidant, so it will reduce reactive oxygen species. And itaconate is a known anti-inflammatory compound in macrophages, as we discussed last week with our guest. And so this does kind of, in a way, can influence the metabolism of the macrophages. And they show that a very important intermediary in this is also mTORC1, because mTORC will the activation of mTORC will reduce the phagocytosis of external or like bacteria. And this is important because there is a dichotomy between having live bacteria and dead bacteria. So dead bacteria seem to be better off. They seem to be phagocyte better and used as nutrients more than live bacteria. And I guess this makes sense because probably that indicates that bacteria might be an indication of kind of a more the end of of an infection, maybe. So you don't want so many activated macrophages. That's how I saw it. I don't think the authors presented it like that, but that's what I thought. Like, oh, it makes sense. If you have a lot of dead bacteria, the thing is almost over. But basically, they show that activation of mTORC1 will reduce this phagocytosis, because phagocytosis results in increased glutathione and itaconate. Then it makes sense that as a consequence, you're going to have the ingestion of live bacteria results in an increase of reactive oxygen species and other things such as IL-1 beta secretion that is also affected by this. So basically, live bacteria increase inflammation and ROS production. And one of the signals that dead bacteria seem to be giving to kind of mediate this recognition is the metabolite cyclic AMP, which is enriched in dead bacteria, and it is converted to AMP in macrophages, and this which results in macrophages that ate killed bacteria have a higher intracellular AMP pool. And one of the things that gets activated by AMP is another kinase, very well known kinase for metabolomics geeks, which is AMPK. And AMPK is known for downregulating the function of mTORC1 as well. So they think that this is a connection by which dead bacteria, cyclic AMP inside the macrophages, downregulation of mTORC1, and this downregulation of mTORC1 results in more phagocytosis of the killed bacteria and the feeding of this nutrient recycling and anti-inflammatory or less inflammatory programming, and that's what they see in bacteria that consume in macrophages that consume dead bacteria. So they recycle the nutrients, they incorporate the nutrients, and this actually affects their inflammatory phenotype to some extent.
So you are what you eat, even as a macrophage.
I know, right? Yeah, that's a very
and they
good.
should go on a diet because if they eat too much, it's bad for them.
Yes. Yeah. So I think that's also the idea that if you don't want to overdo it, if you have a lot of, also, I guess, the live bacteria might be a little bit dangerous. If you want to kind of open the gates to the nutrients, you might be opening the gates to the bacteria themselves. These bacteria, if they're alive, they're also consuming nutrients, so they are affecting the nutrients in other ways. So I think that's also something that will affect the activation of the macrophages. Lack of nutrients activates these metabolic pathways as well.
I can't. There's too much meta here. I'm going to not go down the meta rabbit hole of macrophages, and nope, just not going to. Not gonna. But what we are going to do is speak to Dr. Peter Doherty at the University of Melbourne in just a moment after that amazing segue. But before we get to that, we're excited to be partnering with the International Union of Immunological Societies once again for the IUIS 2025 Congress coming to Vienna, Austria from August 17th to 22nd.
Woo!
Join us. Yes, us.
Yes, yeah. Jawohl.
In your home turf, kind of, sort of.
I know, kind of, sort of. We'll go to the opera.
For a week dedicated to cutting-edge immunology, innovative research, and global collaboration. Early registration are open at www.iuis2025.org. Don't miss chance to be part of the action.
Now, moving on to our second part of today's episode, we have a very special guest for our very special 100th episode of the Immunology Podcast. Yay. We are really happy to have with us Professor Peter Doherty. He's, I think many of us don't need an introduction. I know who he is, but Peter Doherty is, of course, a very accomplished immunologist and co-recipient of the 1996 Nobel Prize for Physiology or Medicine, together with Rolf Zinkernagel. And it might come as a surprise to many of our listeners that we didn't always know how T cells recognize antigens, how they find those infected cells. And there was a time in which we didn't understand what MHC really did with the T cells and how important it was for our own bodies to have, this MHC presenting peptide, presenting antigen to the T cells. And this is the basis, of cellular immunity. So Peter Doherty was one of the discoverers of the importance of the MHC in presenting antigen, or in that moment, he's going to explain better to us how this came to be. But his contribution was for especially for T cell enthusiasts like myself. This is a starstruck moment. So thank you so much, Dr. Peter Doherty, for joining us today at the Immunology Podcast and celebrating our 100th episode. And let's just hear about your experience and your work. Thank you so much for joining.
Well, thanks, Brenda. I'm now in my 84th year, so I'm 16 years off being 100 years old. So I'm probably closer to 100 than most immunologists. I didn't start out to be an immunologist. My training at university, I went to university and trained to be a veterinarian.
Mm-hmm.
In fact, I'm the only person with a veterinary training who's so far won a Nobel Prize. But my interest was in the area of viral pathogenesis. I was very interested in how viral infections work and how they cause disease and damage, and of course, how we recover from them. That's part of it, of course. But I wasn't trained as an immunologist. I was basically a pathologist and worked as a pathologist in Australia and then in Scotland, in Edinburgh, where I first encountered some first-class immunology, but that really, and we made a discovery which impacted on how the immune system works in infection. This was in veterinary models. I was working with sheep. I was a sheep doctor.
That's so funny.
We actually found, well, the thing we found that was interesting is that plasmablasts, antibody-forming cells, were going into the brain of sheep with a viral encephalitis. And they were localizing there, and they were producing antibody locally for some considerable time. That had been speculated about early on, but quite frankly, nobody had actually shown it. So it was a big demonstration, but it was published in a veterinary journal, so it was totally lost, and nobody ever looked at it. And while I was in Edinburgh, I sort of started to engage more with immunology, partly because of that discovery, partly because I'd always had an interest through the writings of Sir Macfarlane Burnet, the great Australian scientist, who was principally a virologist, but got into immunology. And as people did at that stage, if you were in infectious disease, you were right across the whole spectrum, from pathology to clinical disease and so forth. He was a medically trained, and it was a very broad type of approach. So I'd been interested in his theory, thinking about immunology, and I'd read his book on immunological surveillance, for instance, which was about immunological surveillance against cancer. And then, just then, it wasn't only that we didn't know about MHC restriction, we didn't know very much about T cells. In fact, there was still people who didn't think there was a difference between T cells and B cells.
So the year, right? This is the 70s. This is the early 70s. Your kind of seminal papers were published in the 74.
We didn't know what a lymphocyte did until the 1960s with the work of Jim Gowans, who, like a lot of people in Oxford at that time, were draining lymphocytes out of the thoracic duct of rats. But these guys, they were lymphocyte physiologists. Some of them thought that lymphocytes were a kind of a feeding mechanism for the gut. There was no clear picture of what lymphocytes did. And Gowans did fantastic experiments where he used the latest technology. And that's a point we should bear in mind. Our subject, and in fact, medical research in general, advances as much by developments in technology as anything else, because what that allows us to do, and of course, as we all understand, the immune system is immensely complex. And part of that complexity is it moves around the body all the time, and we don't even know how big it is at any one stage. So the technology that allows us to probe better often is what leads to the increase in understanding, rather than any brilliant idea. So anyway, what Gowans did is he radio-labeled the lymphocytes and injected them back in, and then used autoradiography, where you cut sections and you expose film, to show where the lymphocytes had gone back in the rat. And he found they went into the lymph nodes. And so until then, we hadn't even tied lymph nodes and lymphocytes together. And he also did some very good experiments with graft reaction. And then along came Jacques Miller and Robert Good too, to some extent in the United States, but principally Jacques, I think, who showed that neonatal thymectomy, and he wasn't looking for this, he was trying to study a virus-induced cancer of mice. Jacques showed that neonatal thymectomy compromised the immune response in a major way. And that's when the whole T-cell area kicked off. So when I came into immunology in the early 70s, right at the beginning of the 70s, in T-cell immunology, in fact. It was a very new subject, and there wasn't a hell of a lot known. We were talking about helper T cells or and killer T cells. There were good killer T cells killer T cell assays. The cytotoxic T cell assay, which is what we used principally to study alloreactivity, graft rejection. Particularly a group in Lausanne, headed by Jean-Charles Serratini and Teddy Brunner, was doing wonderful in vitro work with the alloreactive T cell response and doing these killer T cell assays. And Zinkernagel, as a young medical graduate who'd done a course in immunology, was working in Lausanne and was aware of these killer T cell assays. And people were also starting to use them with virus infections. And that's where I came into it.
Right. And so you did this work together with Zinkernagel in Melbourne, right? Or Canberra?
No, in Canberra, at
Canberra.
the Australian National University. The Australian National University, at that stage, it was what's called the Institute of Advanced Studies. It was set up after the war, after World War II, and it was the first, and it was set up to award the PhD degree. At that stage, you couldn't do a PhD in Australia. No, people wrote higher doctorates. They accumulated all their work together over a long career and wrote doctoral theses. But the PhD was very much a European innovation. Then the British took it up to some extent, but the Americans took it up in a big way because the Americans had an enormous influx of top European scientists due to our friend in Germany in the 1930s. And so the American science became very, very influenced by senior German scientists, and the PhD became a qualification. Now, of course, the PhD is our research ticket, isn't it? It's what gets us in to science and starts us on our career, in effect. And so, so the ANU was set up to award the PhD degrees. It was postgraduate, funded by the national government. And at that time, there was a very good group there working on the latest in viral immunity with T cells. And the experiments were basically T cell transfer experiments, an area pioneered by British immunologist Avrion Mitchison, the lymphocyte transfer. And basically what had been done, a guy called Bob Blandon had transferred T cells from mice immune to the virus ectromelia or mousepox into mice he'd recently infected with ectromelia and shown that these T cells cleared the virus. And that was about where it stood. Other people were doing these killer T cell assays, and Blandon started to do that with ectromelia through a postgraduate a PhD student called Ian Gardner. And Zinkernagel had been trying to do these killer T cell assays in Lausanne. He came out to work with Blandon, actually. I was there to spend a couple of years. My veterinary, I'd been 10 years out of veterinary school. I'd done my PhD in Edinburgh in pathology and part-time externally in a research institute. I used to go and see the professor of pathology every year, and my supervisor, and tell him what I'd done. He was very nice, morbid anatomy pathologist. And I'd tell him what I'd done, and he would say, well, that's very interesting. You know, I'm untrained, self-brought up, badly brought up, and totally unlike any modern PhD.
Yeah, I feel like it's like a different planet.
I've always run my own research program. I ran research programs from the age of 22 in the state government labs and then in Edinburgh. But I wanted to learn about T cells. I heard a lecture about T cells. I was supposed to come back to Australia from Scotland, Edinburgh, to work in Melbourne with the big government research organisation, the CSIRO, doing veterinary pathogenesis work. There was a big animal health program. We had big animal industries, particularly up north, and they just got into virus infections and so forth. So I was supposed to work with them, and I would have taken a senior appointment there. Instead, I decided to go to Canberra to learn about this new stuff for a year or two, and I took a postdoctoral position in Canberra to learn this new stuff. They didn't expect me to stick around. The guy I was supposed to work with, a man called Cedric Mims, who was a pioneer of virus pathogenesis work, recently died at the age of about 97. Cedric left just before I got there and went to England to become professor of microbiology at Guy's Hospital Medical School. So I was again running my own research program, nobody supervising me. Zinkernagel turned up because he wanted to learn some immunology. He'd been trying to find someone who'd actually take him on. And he had zero interest all around the world. He wrote to all these places, nobody was interested in him, but he met the professor of the Canberra group, the microbiology department there, a man called Gordon Ada, who was an eminent virologist, then immunologist of his time. And Gordon said, well, if you can find some money, you can come and work with us. And so he found a bit of money from the Swiss, and he came out to Australia to work with Blandon. But Blandon's the labs there were very small. His lab was full, and so they stuck him in with me. I was in a tiny box of a lab, but if we could take another person. So we shared a lab, and we just started to talk and do experiments together.
But Zinkernagel, of course, he's Swiss, so he went all the way from Switzerland to Australia to hang out with you, basically.
Yeah. And it wasn't great for him because at the moment, at that time, totally different from what it is now. The Australian dollar was very strong, and the Swiss franc, or whatever it was very weak. So he wasn't very wealthy. Neither of us were, actually. I mean, I've been working in Britain. I mean, in Britain, they pay scientists a pittance, and basically, I needed to rebuild my financial situation, and I wasn't doing that as a postdoc in Canberra. But we obviously clicked, and we started to work together. And I'd become very interested in the inflammatory response, the cells that flood in to a site when you get an infection. At that stage, the people who worked on inflammation were totally uninterested in immunology. They did experiments like painting turpentine on the skin of a rat and looking at the inflammatory response. And so it was kind of unusual for an immunologist to be interested in inflammation. I was interested in inflammation in the brain, and I had a technique where I was getting cells out of the brain and out of the meningeal fluid and the inflammatory cells coming at the brain. I was able to count them. One of the problems with pathology, of course, and particularly with inflammation, is actually quantitating what you're looking at. And pathologists tend to use scoring systems like 1, 2, 3, and 4. But I was actually able to get cells out of the meningeal fluid in the brain of a mouse and count the cells and do proper quantitative studies. But when Zinkernagel came into the lab with me and he started to try and do these cytotoxic T cell assays, I said, why don't we look at these inflammatory cells I'm getting out of the brain and see if they'll kill virus-infected cells? And we did that. There was a little paper published in the Journal of Experimental Medicine early on, and the killer T cells killed like crazy. They were even more potent than the alloreactive ones. So that's what led us down the killer T cell road and led us to compare the difference between different mouse strains, because there was some suggestion that what were then being described as immune response gene effects, which are actually involved with what we now call class 2 MHC, were genetically determined by mouse strain. And so we did an experiment where we compared 2 mouse strains, and we found our killer T cells worked in the homologous system, but not in the heterologous. Just by chance, we were using CBA mice, which are H2K. We were using L cell targets, L99 fibroblasts, which are also H2K. We weren't even aware of that when we did the experiment. And our killer T cells from the CBA mice killed all hell out of the H2K virus-infected L cells. And then when we used BALB-C, I think it was E57 black, they didn't kill at all from the other mouse strain. And so we said, wow, we've really got something here. And we been talking about IR gene effects and so forth, which was a very, very confused area. People were arguing that there was an IR gene effect that targeted T cells, and T cell had two receptors, one for the antigen and one for the MHC. That debate went on until the mid-'80s, actually, until Townsend came along and cleared it up. And Wiley and Pamela Bjorkman and Jack Strominger did the X-ray crystallography. And then, of course, Mark Davis and Steve Hedrick found the T cell receptor. So after we made this initial discovery, which we were then able to map out with all sorts of different mouse strains, recombinant mouse strains, because there was a fantastic technology there already for us to exploit. And that was the technology that had been developed by George Snell at the Bar Harbor Lab. He shared the Nobel Prize in 1980 with Benassareff and Doherty for HLA, H2, MHC, that sort of thing. Snell was the H2 guy. He set out to study tumor immunity and then realized he was studying alloreactivity. And they'd basically made all these recombinant and inbred mouse strains, brother sister matings, to isolate the various loci. Classical genetics, classical back-crossing genetics. And all these mouse strains were available to us, and we were just able to get the mouse strains and just map the thing. And initially we thought, this is one of these IR gene effects, but then we mapped it to class 1. And that was the first time that any immune effect other than graft rejection had been mapped to the class 1 loci. And so we postulated, and this was what was totally original, we postulated that the T cells were recognizing altered self, that the virus was in some way altering the class 1 MHC glycoproteins. And we wrote a whole theoretical interpretation around that. So our contribution was as much the actual experiments as the actual hypothesis around it, which said, look, this is the basis of immunological surveillance, the T cells checking out modified MHC molecules. It explains why the MHC genes and glycoproteins are so polymorphic, because you don't want holes in the repertoire and so forth. We got everything right except the peptide. In a little article we wrote for the hypothesis section of the Lancet. Now, that might seem odd that we would write for that format, but that was the only format of its type around at that time.
Right.
There was no advances in immunology hadn't started. There was a volume called Transplantation Reviews, which then became Immunological Reviews. That was the only review format for large reviews. The very first description, actually, of MHC restriction is in a 1973 issue of Transplantation Reviews, which we'd been commissioned to write by Göran Möller, the guy that had started Transplantation Reviews. Göran Möller was at the Karolinska Institute in Stockholm. The Karolinska Institute selects the Nobel Prize winners. He worked with his wife, Erna Möller. Erna Möller was on the chair of the committee that awarded us the Nobel Prize. And one of the things we did a whole lot of experiments very quickly after this discovery in 1973. Then in 1974, we had a pretty good story together about MHC restriction. We localized it to the class 1 genes, not yet to the MHC mutants, but to the class 1 genes. The second international immunology meeting was held that year in Brighton, and Zinkernagel and I both did grand tours. I went right through the States. I think I talked in about 40 institutions in about 3 weeks or something. It was some enormous amount. I've forgotten the number. In New York, I talked at 2 universities in one day, which they'd never heard of. I talked about this MHC restriction finding. People thought it was important. Most of them didn't get it because they were so used to think of transplantation genes as defining foreignness. And we were telling them that transplantation genes define self. So that was our contribution. I think really our contribution is we discovered the basis of immunological surveillance, that it's targeted to these class 1 MHC genes.
So what made an immunologist care about inflammation to begin with?
I wasn't an immunologist, I was a pathologist. Pathologists studied inflammation, and I was studying, I had got very interested. We were doing a series of experiments on a virus called Laupinellin virus, tick-borne encephalitis virus. It's the only member of the flavivirus group that operates in Britain. It's broadly related to yellow fever virus and West Nile virus in the United States, for instance. And so I was working on that. I had intended to go to Edinburgh. I was a neuropathologist. I applied for it. I was working in a veterinary diagnostic institute in Brisbane. I was required to do that because the Queensland state government had paid my university fees, and I had to work for them for 5 years. I was bonded. It was a mechanism people used in Australia, governments used in Australia, to get veterinarians and dentists and doctors to go to remote rural areas. They got young guys, paid their way through university, and then are required that they work for them for a time. So I did a bit of work in the field as a veterinarian in what Australians call the bush, but then ended up back in the laboratory doing diagnostic pathology. And there was a job advertised in Nature for a neuropathologist, a veterinary neuropathologist. And I was interested it was from a place called the Morden Research Institute. I knew that it was a place you could go to in Britain and that you could do a PhD through Edinburgh University while you were working there. So I wrote off to them and I said, and they wanted a senior veterinary neuropathologist with years of experience. And I wrote off and said, I've never done any neuropathology. I guess I can learn it. I've been doing some pathology I'd be interested in coming there and working and also doing a PhD through Edinburgh University. And I expected to hear nothing more from them. But I got a letter by return mail. That was the way things worked. It wasn't email. And they offered me the job because I was the only applicant. So that's how
only applicant.
Yeah. So that's how I ended up in Edinburgh doing neuropathology. But I thought I was going to work on the prion disease scrapie, which at that stage we didn't know was a prion disease and was very mysterious. And it was a big subject in Australia because the Australians were up in New Guinea studying the prion disease kuru, which was worked out to be caused by cannibalism, ritual cannibalism. And when they stopped ritual cannibalism, it stopped transmitting. So I was going to work on that. But every experiment I suggested when I got there, when I talked to the scrapie people who were going mad with frustrations because they were trying to deal with it as a virus, every experiment I suggested, they said, oh, we tried that. The experiment took us 7 years and we didn't get a result. So I thought that wasn't a promising PhD project. So I switched to working on this viral encephalitis, which was problematic at the moment because though they had a vaccine, they were getting a lot of infections in people working with it, and also the vaccine wasn't working that well. So I worked on the pathogenesis of that. That's where I got very interested in inflammation. That and from the previous work I'd done in Australia with a kidney disease called leptospirosis, where again, I got the impression inflammatory cells from looking at antibody levels in urine and blood. I got the impression that antibody cells were flooding into the kidney and producing antibody there. So that's what got me into inflammation, basically. And I was doing electron microscopy, histopathology, post-mortems in the middle of the night, often when these sheep would get sick. And I was working with another young guy who just died recently, who we shared the same sheep for our PhD theses. I did the pathology, the experiments, I ran the experiments and the electron microscopy. And Hugh, my colleague, did the virology and the antibody measurements. And so we worked closely together. For a time, I wasn't doing much because I jabbed myself in the finger and gave myself the infection.
Oh.
So we got the local doctor to come along. I took my temperature, She bled me every day. Hugh looked at the samples and isolated the virus back from me and looked at the onset of the antibody response. So later, I didn't get sick, fortunately, though some people got very sick with this virus. Later, I looked at the data. There were also a few other people infected that were also studied. I looked at the data. I wrote up the results, put the medical practitioner on as first author, and we published it as a letter to the editor in the Lancet. So it's the only time I've been an experimental animal.
Well, you're joining the ranks of so many illustrious scientists experimenting on themselves, I guess.
Not intentionally, not intentionally. And I think, I might have been quite smart if I hadn't had that neurological disease, but it probably dropped my IQ considerably.
I have to say this. It's such a long career, so many things
Yeah,
you did.
sure,
So
very long.
let's talk about the life after the Nobel Prize or after your discovery. So I'm pretty sure you knew when you published
Yeah.
those Nature papers in '74, you knew this was big and this was going to be very
Yeah, I
influential.
mean, basically the format, they weren't Nature papers. The Nature format at that stage was one that I absolutely loved. It was about, it was the Nature letter.
Oh, the letter.
I think it was maximum of 1,000 to 1,500 words
Yeah,
and
it's very short.
4 displayed pieces of data. And this suited my personality totally. I like to write. I write very concisely.
Right.
This was a time when, if you published in the Journal of Experimental Medicine, if you look at the papers back then and earlier, you will find, and the Journal of Experimental Medicine was the leading journal that published immunology out of the Rockefeller University. Cell, Nature Immunology, Immunity, none of these journals exist. There was just one Nature journal. That was it. Okay, so this very short format, and we published, I think, maybe 4 to 6 of these Nature letters. And that's how Watson and Crick published their findings. And actually, the lady who died, Rosie Franklin, had her paper in the same issue, again, a letter to Nature. And I mean, I've always admired that letter because it sort of describes what they found. And at the end of it, the very last sentence, have you ever read the Watson and Crick Nature letter?
Uh,
If you haven't,
yeah, but I
read it,
remember
because the very last sentence is something like this. It has not escaped our attention that this explains everything or something to that extent. It explains genetics. And that was their very last sentence. Typical British, understated in the very last sentence. They actually say what it's about. People didn't realize what they'd done for quite some time, actually. A lot of people missed it. And it took quite a while for Watson and Crick's discovery to catch on. So I had learned in Edinburgh, I'd learned to write very, very concisely. I don't know, but if you know, but the Scots have a reputation for being mean. They're not mean, they're just parsimonious. They don't waste words.
Fair enough.
And so I learned to cut my writing right back and to be very, very concise, which is very good for scientific writing, not necessarily so good for other types of writing. And so I could say a lot in a very short space. And it suited me perfectly because now if you send a paper to Nature with speculation in it, they say, well, they can do these experiments. They'll demand lots more experiments.
Yeah.
And of course, at that stage, you couldn't do the experiments, so you could speculate as much as you like, and nobody could ask you to do the experiments. There was no online data, and so perfect.
Exactly.
That's my style. I wouldn't make it as a scientist now.
Golden days. I mean, I feel like I'm being that I was born in the wrong time. 17 figures in the supplementary to get it through, but
It's terrible. I think it's terrible. I think the papers in the main are almost unreadable by the time they're so re-reviewed and rewritten.
yeah.
You need a Nature News and Viz to explain what the hell the paper's about. And
Indeed.
I actually, and also I think people should be able to speculate, and they should be able to put stuff in a discussion that's not backed up by rigorous data. I think these papers are over-reviewed. I don't think the editors largely know what they're doing, and they rely too much on reviewers. They need to make executive decisions and make these papers more interesting.
I agree, and that would get it out of the political hand, right, of the other reviewers, because some reviewer knows that they're reviewing your rival's paper. They're going to say, oh, I'm going to give them another 2 years of experiment.
People do bad things, quite frankly. It does happen. I don't think it's the main, but it does happen. And I know my molecular virology colleagues were always much more worried about it that much earlier, because it was possible to do those experiments so quickly. A lot of the immunology experiments of my time, at least, were so complicated that it wasn't really easy to actually do them. I mean, me studying inflammation in the brain of a mouse, and we did all the MHC restriction stuff we did in vitro with the cytotoxic T cell assay. We did in vivo with transfers, immunological transfers. Papers tended to end up in things like Journal of Immunology. And I know at least one of them was reviewed in, and I heard this later, it was reviewed by the Benacerraf group in Boston. They discussed the paper widely at the review stage and then rejected it. There were some funny things going on, actually. Immunology became a very silly subject for a while. I mean, do you know the whole suppressor T cell story from the 1970s, which turned out to be absolute nonsense, actually, and didn't go anywhere. It's not the same as the regulatory, the Tregs. I mean, that's a different story. These were supposed to be CD8s. And if you looked at, I mean, I don't know whether they still do it, but the phenotyping from clinical labs at that stage described CD8 T cells as suppressor T cells. They're categorized as suppressor T cells. And a whole generation of medical doctors who were trained in the 1970s were brought up on this stuff. And it's wrong. Of course, CD8 T cells are suppressive. They kill. That's very suppressive. Being dead
ultimate suppression.
But it was nonsense. And it was bad science, badly done, and taken far beyond what the data was actually saying. I don't sort of go on about it much, because nobody needs to be writing negative stories about science. I mean, there's enough negativity out there, but it was not a good time in immunology. Immunology was cleaned up by molecular biology.
Yeah.
There's no doubt about that. It's a very messy subject, very, very complicated. We've got these two enormously complex systems, the brain and so forth, and the immune system, both of which are concerned with reacting to input from the external environment. The brain, at least you know where it is. There's no central processing unit in the immune system. One of the questions I'm still obsessed with is how does the immune system count? How is it that we have relatively stable CD4, CD8 ratios in the blood? How is it that we get these enormous clonal expansions? And we saw this with influenza, for instance. Some of the epitopes will give you enormous clonal expansions, and some will give a very limited clonal expansion. But within months, the numbers of memory T cells can be, in blood at least, can be back to much the same level. So there's they must count in the lymph nodes somehow, but I'm not sure how it happens. Maybe we're getting more insights now with these wonderful in vivo imaging techniques and some of the fantastic work people are doing. I mean, immunology now is fantastic. I mean, the power of it and the way that it's moved into clinical medicine in such a dramatic way. It's very expensive medicine, unfortunately, but I mean, it has given dramatic results. If you talk to cancer specialists 15 years ago, they didn't even want to know about immunology. It wasn't any help to them. They weren't getting anywhere with it. And then along comes Jim Allison and then the monoclonal antibody therapies and so forth. And we have immunology right at the forefront. And those guys have done great stuff. And bringing things forward, and it's been a great human benefit.
So I was going to ask you if you could solve something, what would be next? But you answered that. So instead, I'm going to ask you, got a Nobel Prize. Lots of people have been interviewed. Okay, you get the call in the morning. Do you think it's a prank or not? All of that. But what about, the weeks and months and even years after that? Like, how did that go for you? I mean, I'm sure in some ways nothing changes and yet everything changes. But for you,
Yeah, well, actually, at that stage, I'd been working. I'd been in Scotland and then back to Australia. After we did that groundbreaking work and made that big discovery, they couldn't get rid of us quickly enough from the Australian National University. I think the thinking was that, and they'd had this experience before, that Zinkernagel and I would go away and disappear, and they'd really take it on and drive it forward. And of course, neither Zinkernagel or I are the sort of people who elect to disappear. We're both loud and we both keep going. So I went to Philadelphia for 7 years. I was at the Wistar Institute for 7 years. I ended up being I was an adjunct professor at Penn, and I had graduate students through Penn. At that stage, Penn wasn't at its best stage, but now, of course, it's one of the absolute top universities again. The trustees of the University of Pennsylvania had invested much of their endowment in the Pennsylvania Railroad, which then went broke. And so Penn wasn't in great shape, but now it's a great university and it's doing great stuff. But it was a great experience working in that environment. I was in an independent research institute, came back to Australia. That didn't work out too well. So by the time the Nobel Prize came along, I was head of immunology at St. Jude Children's Research Hospital in Memphis, Tennessee. A great institution and a very unlikely place. I mean, but incredibly well funded and with some really top people, a number of members of the National Academy of Science and so forth, and a public fundraising campaign that raises $1 billion a year. So, substantially funded, if you like. So I was in Memphis, but I was Australian, and I wasn't an American citizen at that stage. At that stage, if you became an American citizen, you had to give up your Australian citizenship, and I wasn't willing to do that. So I was there on a green card. And when the Nobel came, I actually elected to be with the Australian contingent. That is, what happens when the Nobel happens, all the prizes, apart from all the science prizes and the economics prize and the literature prize, are all awarded in Stockholm by various committees of the Swedish Academy of Science, the Academy of Literature, or of the Karolinska Medical School. The Peace Prize is awarded in Oslo by the Norwegian Parliament. In Stockholm, the ambassador of the country where the Nobel Prize is going to come from then has quite a prominent role through the whole Nobel thing. And so we were with the Australian ambassador, not with the US ambassador. And then pressure was gradually put on from various sources, including the fact that we didn't want to die in Memphis, to come back to Australia, which we did with 1956. We came back to Australia 8 years later. Still had worked part-time in Memphis for another 10 years or so, but we're mainly based in Melbourne. So the transformation that happens, I hadn't been on the public stage much at all. The Nobel Prize puts you on the public stage, and everyone in that year after the Nobel, in the US or anywhere else, particularly in the US where there are a number of Nobel Prize winners, you're the sort of person that everyone's trying to get for their named lecture and all that sort of stuff. So you have a very busy year. The institution actually appointed a media person as my minder for a year to organize all that and look after it. And then it can calm down. But I found that I had a facility for public science communication, and also just as I was really back from the Nobel Prize celebrations in December 1996, I got a call saying I'd been made the Australian of the Year.
Oh, that's quite an honor.
Yeah, well, it was, because I didn't even know what it was. And they said, well, what am I supposed to do? And this is December, and I've got an incredibly busy year ahead of me with the Nobel thing. And they said, oh, well, you have to come out to Australia and you have to talk in all the capital cities to public audiences and be interviewed. And I said, well, could I put it off for a year because I'm rather busy next year? And they said, no, you can't if you ever want to visit Australia again. So I came out to Australia that year as Australian of the Year. We made a number of trips out, actually, and I discovered I was good at public communication. I didn't really know that, but I've got some Irish genes, and as a result of that, I can talk incessantly and underwater. And so, so I sort of fell into public science communication. And since then, beginning in 2005, after I'd returned to Australia, I've written 7 books on science and the scientific life,
Yeah.
and done an enormous amount of writing which is largely wasted for magazines and newspapers and stuff. And also written a book on war and tennis, but that was a different matter. But so I became a public science communicator. But all that time, after the Nobel, one very big thing happened, and that was the development of the tetramer technology by John Altman and Mark Davis. And that meant we could both quantitate responding T cells and do single cell sorting and look at their molecular profiles. And that's what revolutionized T cell immunology, the tetramer technology. And we did really terrific experiments over the next 10 and then 15 years with some of my younger colleagues really substantially taking a lead using that area. And of course, that's what's transformed human T cell immunology, because being able to do single cell work means you can work with very small numbers of cells, and you can work with, say, pediatric populations, which were pretty inaccessible when you needed large numbers of lymphocytes, and you can work with the elderly and all the rest of it. And so that's been enormously transformative. And we played a pretty significant role in that for some years. And then that's been passed on to my former postdocs, all of whom are now full professors somewhere or other, and driving the whole thing forward.
A great legacy. That's I think something you can be very satisfied with.
That's science. I mean, Nobel Prizes, they're kind of a celebrity thing, but science isn't a celebrity culture, and it shouldn't be. And in fact, the Swedes worry about that, but then they're not going to abandon the Nobel Prizes because it gives them a lot of clout, actually.
Yeah.
This is the prize everyone takes notice of.
Although, if I may say, it's a little bit rich coming from someone that literally has an institute named after himself. So
Yeah, that's right. Well, I tried to get them to find a very rich guy and give them a lot of money and name it after them, but we didn't find one. So they called it after me. And I must admit, it makes it very hard to escape. And everything that the Institute is, through COVID, of course, we were very, very prominent. And the Institute, which is a new institute, only 5 years old, came to major prominence globally because we had a unique structure where we had the academic immunology department, microbiology immunology, very good department with very good people in it, like Bill Heath and now Laura Mackay and all these sort of people. That was combined into this new institute and the training, and it trains a lot of younger PhD students, with the state virus and microbiology diagnostic labs and the World Health Organization Influenza Center, which were all on separate sites, and they all came into this new institute. So when COVID hit, we had both the basic science and the diagnostics,
right,
and the network of the influenza network, which is the best established infectious disease network, apart from, say, maybe HIV/AIDS. And so we were in a unique position, and our institute was the first outside the US to isolate, outside China, to isolate the virus. And not only that, the Chinese were sitting on the virus, and we gave it to everybody who wanted it straight away. And so, we first became aware of COVID in December 2019, Australian Eddie Holmes, a molecular guy who works on virus diversity, pushed the Chinese to publish the sequence. And they published the sequence, I think, in mid-January, something like that. Not totally with the approval of the Chinese government, and some people got into trouble. But it was out there, which meant people could start developing PCR tests and stuff immediately.
Yeah, that was very important right at that time, the speed.
By the
of
time
that.
we got our first case, which was 24th of January, I think, a guy coming in from China who knew exactly what the problem was, very responsible, self-reported to one of the major universities, and was treated in isolation right from the outset. We isolated the virus from him, and we already had a PCR test up and running that was perfect. And so we were right out there very, very quickly. And with that unique structure, we're very prominent. But as a result of that, we get blamed for everything by all the cookers and anti-vaxxers and nutcases that are infesting the planet. And we got money from TikTok, for instance, and Jack Ma from China. They gave us money to work on the thing. The idiots interpret that as that's being given to the scientists personally to enrich them, and that we're working for the Chinese Communist Party. At one stage, because we were also pushing vaccination, I was accused of working simultaneously for the pharmaceutical industry criminals and the Chinese Communist Party. So I was very conflicted. I never got a cent from any of them.
Strange times.
It's insane, and what's happened politically in the United States is insane. I mean, it is crazy.
Jason, you don't see it, dear listener, but Jason is like laughing nervously at the accusation
of the swing state.
Yeah. You ruined it. You Pennsylvania, you ruined it for everyone.
It was a phenomenon that I think we haven't learned how to handle, and I don't know if we can handle it. We haven't learned how to handle the dissemination of false information and propaganda via the internet, via social media. And, I travel by tram a lot in Melbourne. We have trams and we live centrally. And I sit on the tram, and when I came back to Australia in 2002, you'd sit on the tram. A lot of people would be reading a paperback book or something because, it's boring and it's a way you can just sit. If you could get a seat, you'd sit there and you'd read. Now they're all looking at their iPhone and they're sending out important messages like, hey, I'm on the tram.
Yeah, there's a lot to unpack there. I also remind people in the rest of the world that no incumbent has ever won in the U.S. after having the inflation that the U.S. had. They always get voted out every single time, swap parties.
Is the economy stupid?
The fact that it was so close in the end shows how much of there was a pushback. But in the end, it wasn't enough given the inflation. Like, you have the inflation numbers, you get blamed, you're out, and it still was ultra close.
We may have the same result here very soon in an election. But I think, everything that Trump is proposing is going to be highly inflationary. So it's going to be interesting to see what the reaction is.
We'll see. Okay, guys. I think we should maybe stick one more question that we want to ask you, Peter, because I think we have taken a lot of your time, and it's been such a fun, interesting conversation. There's a question we like to ask our guests in the podcast. That's kind of a wrap up. And it has to do with, who are they outside of their science or like outside of the work that we discuss. And we really like to ask that if you were not, had not ended up being, a successful Nobel Prize winning immunologist with an institute named after you, and a legacy, that spans many, many, many people, what would you think you would have done? Is there something else that you would have liked to do if it wasn't for your scientific career?
Yes, I mean, I think I thought about a number of things when I was finishing high school. I had a kind of odd high school experience, public high school, but it was a school. I was in the first year of students in the school, was a brand new school.
Oh, that's
And
fancy.
they just popped this school on the side of a hill. State government provided some very good teachers and a building, a bit of science equipment. But there was no library and no clubs, none of the things you associate with a high school, and basically no older students. And not only that, I was also a year younger than most of the kids because I'd been pushed up a year as a kid, a little kid. And so it was a very odd high school experience. And it made me, I think, on the one hand, very independently minded, but on the other hand, very disrespectful of authority, which has been my characteristic, which gets me into trouble. I mean, that's why I haven't taken big administrative jobs, because I always can see what needs to be done, but don't have the patience to spend 10 years trying to bring other people with me. But it was an odd experience. So at the end of my high school thing, I was thinking of, I had an older cousin, 13 years older. My parents and most of their generation, I was growing up in a working class, lower middle class area. My parents' generation had largely left school at 15, as my parents had. My mother was a music teacher. She'd trained as locally and was actually qualified through Trinity College London as a music teacher, piano teacher who hated to teach and liked to play. And my father had trained as a telephone technician, and then with the then government-controlled telecom service, had worked his way up through that doing internal exams and was kind of at an administrative level in that. So I had no experience. We were my generation was the first generation in the family who went to university, which was very common at that stage, much less common in the United States and in Europe. And I had an older cousin, 13 years older, who'd graduated in medicine, a brilliant guy at school, and then he'd got into infectious disease, trained, done a Master of Public Health at Harvard. He's the guy who isolated the Ross River virus, which is, I don't know whether you know virology very much, but it's basically very similar to the chikungunya virus, which causes polyarthritis and rash. There's a big immunological component to it, which we don't understand. But he later went off into academia and social science more than basic science. But I thought in terms of doing research, but the only medical doctors I know sat around apart from my cousin, in suburban offices listening to people whine about being sick, or that was my perception as a 16-year-old with zero empathy. And I didn't want to do that, so I decided to be a vet and work on of food-producing animals, because at that stage I was quite religious and embedded in Methodism, which is a bit like the Calvinist. What would you say? The Netherlands is Calvinist, or
Protestant.
Well,
Calvinist.
Edinburgh's Presbyterian. So Methodism's kind of an offshoot of the lower Anglican Church. And at that stage, they didn't drink and they didn't dance.
So neither did you.
No, not until I got to university, when I sort of moved on. But basically, so it was odd. So I was good at writing. My favorite subjects were history and French. I never met anyone French, but there were no French people around. But we classically learned French in schools along the English tradition. And it was my first introduction to another culture and another language. And if you learn another language, of course you're very familiar with this in the Netherlands. How many languages do you speak?
Well, people in Netherlands usually speak at least 2 or 3. 3 is fairly common.
Speak 6, right?
I speak more, but I'm not from the Netherlands. So I speak Spanish is my mother tongue. I'm from Argentina originally.
Okay.
And then English, German, some Dutch, and then I understand Romance languages in general, like Portuguese, Italian.
And the American college system, I think, pretty much forces you to learn a second language, doesn't it, Jason? Or it used to. I know.
Yeah, it mostly does. And how well you retain that depends on how much you use it afterwards.
Well, I mean, I met a whole lot
Jason speaks Spanish.
Try, and you mock me.
I do. I'm sorry.
I've met a whole lot of Americans who are educated through very expensive colleges, which are fantastic educational institutions. American higher education can be fantastic at the top end, both with the liberal arts colleges and with the major universities. But I also, having lived in the American South, I've met many people who've had fantastic educations. And as I think of it, they escaped completely unscathed. Their mind was not changed on anything.
But maybe that was the education. Make sure that the mind stays where it should.
Yes, I think they're embedded in belief and conviction, not in evidence. So I thought about a career in journalism. I considered applying for a cadetship as a journalist at the local newspaper, which ends up being which is a terrible newspaper. It's one of the worst newspapers in the world. So I thought about that. I thought about going to the National University, which at that stage, we hadn't engaged with Asia until the Second World War, which, as the Japanese came down very close to Australia and bombed Australia, and my two uncles, I wrote a book about it, this book about war and tennis, were involved in fighting in Southeast Asia against the Japanese. We realized after the Second World War, we had to engage with Asia. So the Australian National University had set up a school of Asian languages and Asian culture. They had very good scholarships. I thought of applying for one of those, but didn't, but ended up applying for this scholarship to go to the veterinary school, which is a weird choice because I was a city kid. And if I'd ever worked with a vet, I wouldn't have probably done it, but I didn't. Anyway, so I've got this weird career. I'm a failed vet, basically. So.
Well, but so journalism would have been an interesting option, but you kind of already are using the spoken word, and you're doing a lot of communication and a lot of writing.
I am a writer. And basically, if, Rolf came out of, Rolf came from, his grandfather was a professor in Germany, and his father was a very, very prominent scientist at Hoffmann-La Roche. In fact, Rolf felt he should have shared a Nobel Prize with, I forget what the discovery was, but he didn't. It went to the clinician. So, he came from a very well-educated, typical upper bourgeoisie Swiss family, and with the various attitudes and culture that goes with that. And but we hit it off fine because the Swiss are pretty skeptical and Australians are very skeptical. It's a bit because of the climate, that when Americans came to America and they farmed land that had a soil depth of about 3 meters and which had nice steady rainfall. That's changed a bit lately. In Australia, people came and farmed land which had a soil depth of about 10 centimeters. And, it would rain well for about 5 years and then not rain for 10 years. It's a very dry country. So Australians are a bit cynical.
Flora and fauna also want to kill you.
Yeah, everything wants to kill you.
Want to kill you in Australia is the joke, at least in the States, is like you guys got like the most poisonous everything and these crazy spiders that are the size of your head and kangaroos are violent.
The spiders that are dangerous aren't very big.
But you have the tiny deadly ones and the ones that will eat your pet.
But try this one. We don't, people rarely die from spider or snake bite, quite frankly. They die from
rarely.
Well, we don't have the experience where, for instance, a group of people are riding along on bicycles through the California hills and the last one is eaten by a mountain lion. We don't have any bears.
No, but they're like knocked out by a kangaroo or that I've seen. I've seen videos. I've seen videographic evidence.
One of my friends was badly beaten up by a kangaroo, but that's unusual.
Well, we're running out to the end of our time here. So we just want to take a moment to thank you so much for coming on. I think this is probably like the 1 hour that all three of us are available in a day when we record this, given how many time zones we're at. So thank you very much for coming on. I'm sure our guests will love this for a special 100th episode.
Absolutely. This was such a great conversation. It had everything, the science, the history, the anecdotes. This was brilliant.
Basically, science is a continuum. And, some people get get hit with the star system occasionally, but really, there's just incredible people in science. And many of my colleagues, I think, are better scientists than I am, but they haven't won Nobel Prizes. They didn't get lucky.
So with this, we thank so much Professor Peter Doherty from the University of Melbourne joining us today to talk about his work, his legacy, and winning the Nobel Prize in 1996. That brings us to the end of our show. Don't forget to subscribe to our newsletter at immunologypodcast.com to get show notes, including an episode summary and links to all of the interview and Roundup Papers. You can also reach out to us on X at Immunopodcast or via email at info@immunologypodcast.com with feedback or to suggest guests. See you next time.